university students or university staff, and all university students or staff have to learn English as a base. Of course, if you want to run a good program at a university anywhere in the world, you need to be capable to read all the literature that is relevant for your work. Why do I stress this at the beginning of my lecture? I have a case right now going on which is related with my most favorable fish. Like here, Apistogramma is a genus of cichlids that are pure neotropical, where someone has done a revision of these fish. Really nice work, well done. Well, except one point. The knowledge of literature in that case was, oh, you want me to, you can hear me? Yeah. So I leave this because usually I'm loud enough. The poor knowledge of English literature led to a very bad situation. In this case, 2 species have been described for the second and third and for the 4th time by one researchers team. They worked out the species descriptions on one species 3 times. This would never have been possible if they knew the literature. Right? Okay, that's the point why I stress it so much. Okay. Today, climate change is a topic in science worldwide. You here experience it right now. Of course, the climate is a little bit changing. And today we had sun in August in Lima, which is, as far as I have been told, pretty exceptional. Usually it should be a little bit more foggy and cloudy. And climate effects are usually seen as effects on the polar regions where the polar caps are melting, and nobody cares about the tropical regions as so hardly being influenced because, well, it's warm there anyway. And what usually is not being seen, some aspects of climate change that are not obvious, especially if we are looking on the biodiversity. And one region which is really exceptional for its biodiversity are the Amazonian lowlands, especially that of Peru. If you go, for example, to Loreto, you have a concentration of species there that is exceptionally worldwide. There's only one area that may be compared to it in Asia and a very small spot in Africa, but concerning to an area, the average richness of species in Peru is exceptionally high. I will try to show you some aspects during my lecture that may be a little bit surprising, but I guess they may influence you in just looking on the things a little bit different, maybe on your research. Here we have a map just plotting megadiversity areas worldwide. Here we have the coastal strips of Australia, everything. And what I mentioned here, this area is unique. And for example, the African lakes, Lake Victoria, contains about 1,000 species of cichlid fish. That is more than we know from the Amazon. Well, but this is an artificial effect, of course. We have started to work on the biodiversity, for example, of cichlids, well, 10 years ago. And the number of species the taxonomical work produces it, the number of species is exploding now. We have been in the field for about 4 weeks, and these 4 weeks have produced more than 15 new species of cichlids just by going there and picking them up. The number is much higher than estimated right now. The examples taken by us here are just important because biodiversity usually is seen as important for business purpose and things like that, but some aspects are not so clear. I have mentioned it in the beginning. Potential impact on the environment is not restricted to the polar caps. So here, this is a look in the Andes right here next to the door. Even here you can see it. I have read a little bit about the problems this city has with fresh water, drinking water. If it's continuing like this, there will be bigger problems in about 2 years, I guess, here in the city, because the supply of fresh water is limited because there is no sources anymore if there is no ice in the areas. But climate change effects are also seen in the rainforest. For example, the last 2 years have brought the highest water levels and the lowest water levels ever measured in the Amazon region around Iquitos. For example, in the Pacaya Samira Reserve, the average water level was about 2 meters higher in the extreme than normal. This year, the water level in end of July had reached the low level of end of August last year, and will probably end up with another 2 meters less than normal, which means the river is pretty close to dry. Last year, river dolphins, manatees, have been trapped and died because they were locked in some areas in the Pacaya Samira, and well, just died because a dolphin without water is a dead dolphin. The species the diversity in the cis-Andean lowlands is super interesting because there are some groups of fish. I will especially have a look on one group during the lecture. But here are some examples. Catfish. Catfish have a diversity of species that is unique. At the time being, we know of more than 2,000 different species of catfish in the Amazon lowlands. Amongst them, a group of about 20 species, maybe 30 or 40. This is a question of the taxonomical view and modern genetical investigation there. That is of highest influence and importance for human consumption. Pseudoplatystoma, for example, Brachyplatystoma, the big catfish, which are about 2 meters big, produce a lot of food for men in this area. Well, a lot of them have been fished down from the food chain. The big ones are lost. Meanwhile, on the markets, you just get 30, 50 centimeter fish of about 1.5 kilo of a species that might get up to 20 kilos. There is other examples. The loricariids, for example, the smaller catfish, have a diversity that is very interesting on another business aspect. Loricariid catfish are trade goods for the aquarium fish industry. Every year, about $20 billion are made by just selling fish from the Amazon. About half of it by catfish, by loricariid catfish. A catfish like this here, I guess it's butterfly catfish. I don't know the scientific name because it's, I guess, not described yet. A fish like this may cost about $1 here, but if you have it in the trade in the US of America or England or in Germany or in France, you have to pay approximately through $300 for one fish. So there's a lot of money that is made by these fish. And this is only one example. You have characins, very interesting. Some characins, you all know, are big and are for human consumption. And there's a lot of species that is interesting for the aquarium trade. Why do I stress this? The aquarium trade is very interesting because a lot of fish from Amazonia are exported to Europe. And in Europe, there is a very intense scientific scene monitoring Amazonian fish in research programs about their reactions to thermostress, to stress by temperature, by high temperatures or low temperatures, and the reactions on reproduction. I myself have run a lot of research on that. I will come to that later. Amongst the characins, there is some that are really colorful, and some of the smallest vertebrates worldwide. That species that I just put in here is in adult size 9 millimeters long. An adult, like this. That's it. You'll never see a bigger one. And you have the big group of cichlids. All in all, in the neotropics, at the time being, we have a rough estimate of about 10,000 described species and probably 25,000 species awaiting description. So it's a very important group of vertebrates. If you look on birds, for example, the number of bird species worldwide is about roughly 10,000 species. Mammals, well, here in the neotropics, bats are very important. You have about 1,000 species of bats. That's exceptional. But if you look on the on cichlids, for example, here we have a cranicicular relative. It's a Batrachops species, very specialized group of comparatively big cichlids living in rapids. We have about 30 species of Batrachops at the time, and about 500 species of Cranicicla, which you usually call Anchoa. About 500 species. On the tour that we have just completed, we just discovered a new species about 10 centimeters long as adult. We just walked to one pond, just picked them out, and it's a new species. So that is typical. Cichlids are perfect models for research, and especially for research on climate change. There's two reasons. You can get them easily. They are all over the neotropics, and you have them in Africa. 2 continents, you have it. And there is one species in Asia, the rest has to live without cichlids. And on the other hand, they are very easy to handle. You can transport them very easily. Very few of them die during transport. You can bag them. You put them in plastic bags and transport them all over the world. And you can put a cichlid in a bag and keep it there for about 2 weeks without opening that bag, without feeding it, without anything. Put it in a cardboard box and carry it around. It will survive. They're really tough. Super for research. And therefore, we of course take cichlids. Well, basically, you have to know what a cichlid is. Cichlids are pretty easy to define. there are fish that are characterized by one nostril. Usually most fish have two openings here, nose openings, olfactory openings. But cichlids have only one, except one species that shows us, this is an ancestral marker, that there is two openings. And the lateral line here is interrupted right here and continued here. In all other fish species, the lateral line is one unit. In cichlids, it's 2 units. And you have hard rays in the dorsal and in the anal fin, and one here in the pectoral. If you see this, you know you have a cichlid in front of you. Super easy. If you have to describe other fish groups, it's very difficult in some cases. And the size of cichlid is pretty good for research because they are usually between about 4 centimeters and 1 meter long. The biggest cichlids are Cichla monoculus, which is about pretty close to 1 meter, the ones from Colombia. They're very, very, very big. And the group of the cichlids has different aspects. You have large ones, you have medium size, and you have dwarf cichlids. Dwarf cichlids are usually not a biological unit, not a biological entity. They are just an artificial effect because we just sample all fish that are cichlids that are smaller than 10 centimeters in average, maybe 15 in Crenicycla, in this. And you find this heterogeneous group all over the Amazon lowlands, and in the Orinoco and in the Paraguay system. As you can see here, you have very different morphotypes, round ones that are obviously have a very small mouth, they're picking insects or detritus. You also have bigger ones with a pointed big mouth. For example, that one in the middle is specialized to live in rapids. They have gone into about every area that you can imagine in the Amazon lowlands and are living there. Some of them are stinging, are only living in extremist type of environment. For example, these two species here on the right side below are living in water, regularly having pH lower than 4, a conductivity less than 10, and temperatures usually less than 25 Celsius, which is pretty low for the Amazon rainforest. On the other hand, you have the one here on top that you can find nearly everywhere. pH 7 to 8 is for them acceptable. You also can find them in white water, black water, clear water, everywhere. So you have specialists and generalists. And that is pretty good because amongst these, there is one group among these dwarf cichlids that is the top group in research because it's Apistogramma. It's not clear why they are called Apistogramma because it's just 100 years ago when the name was coined. But Apistogramma have a few things in common. They are usually smaller than 8 centimeters. They are sexually dimorphic. Females and males are looking completely different. And there is a big number of species and a very high degree of heterogeneity. Well, I have to put some correction in here. Presently, 70 described species is wrong. Presently, we are at 83. Well, this is 1 year old. 1 year in taxonomy at the time being in Apistogramma is very, very long time. And well, 172 undescribed is also wrong. We are pretty close to 300 now. And so this is an enormous, interesting group. If you look here, you have very different morphotypes, very, very different morphotypes. This here are all male fish of 6 different species. The females, well, the males are pretty easy to distinguish, but the females are looking very much different. And we will come to it here. First, the ecology of this fish is pretty well known. We know pretty clear where are they living. The distribution of the species is pretty well known. Secondly, we know from nearly all species what type of habitat they prefer, what water chemical background and physical background they prefer. This is very important when you want to run experiments in aquaria with these fish. They are distributed here. This is some sampling plotted in a map all over the continent, except west of the Andes. The distribution is usually in areas lower than 300 meters. And well, you could say, well, the distribution is a little bit irregular. Yeah, it is, because this is not a map of Apistogramma distribution. This is a map of research activity. We only know things about this species, of course, when we have been there. In between, this area here is completely red. We cover it completely meanwhile, and have a lot more information there. Next year, we will just put this in red, because there will be a research program in Bolivia and in some parts of Brazil and in southern Peru. So, you find Apistogramma in the Orinoco Paraguay system, in the Essequibo system, which is pretty isolated from all others, except that there is a junction between the Essequibo and the Rio Branco, which is part of the Rio Negro system, just like the connection between the Orinoco and the Rio Negro. So we have 2 connections there, and as well a connection between the Rio Paraguay and the Rio Madeira. So it's clear why these fish are in these areas. These are just the confirmed sample sites. And the ecology is very interesting. We have a lot of species that are preferring black water. Black water is just like Coca-Cola. I hope you all know it, and I've seen it. And we had 78 species that are specialized on living in black water. We have 75 species that are specialized living in white water, and we have about 41 species in clear water, but interestingly we have a few species that are able to live in mixed waters. These are the real generalists. Why do I stress this? You have heard about the Wallace line and about Wallace's barriers. Big rivers may have functions as Wallace lines. They are barriers for distribution of animals. From the Amazon, it is well known that you have north of the Amazon, you have different species of mammals than south of it. It may be species, it may be subspecies. You have the same effect on butterflies, you have the same effect on birds. And what is less known, you have the same in fish. If you look on the distribution of fish in a river system, it is not random what you find. You can predict, you can foresay what you will find in a river if you know the ecological background of this species here. The interesting thing is that usually the conductivity is low and the pH is less than 7. It's usually acid water, with some exceptions in the wild. This makes it easy to handle this fish in experiments, because you just have to produce one standard water and you can run this fish there. To know a little bit more about the type of habitat they use, you have to go in the field, of course. And Apistogramma are very interesting because most of the species share an addiction for leaf litter, for leaves that are lying on the ground of the river. They hide under these leaves, and they make use of these leaves in very different kinds. They are grazing, scraping the Aufwuchs, as it is called, micro invertebrates there. They just graze it from the leaves. They just pick up detritus from there, and they lay their eggs on the lower side of the leaves. So they use it as a shelter for their offspring, as well as a shelter for enemies, predatory species like kingfishers or other fish like bigger catfish or some grain cichlids or things like that, or bigger kerosines. Topias is one of the big predators for Apistogramma. And we have 100 species amongst the ones that we have researched that are specialized on living in leaf litter. If you put them in an aquarium without leaf litter, they really do not feel well and die after a few days. We have about 10 species that have only been found in drifting meadows. We have Iconia or Pistia or something like that drifting on the Amazon, and you collect in there, you find specific species of Apistogramma there that you won't find at any other place. We simply don't know how they managed it, but they do it. They are there, and it's regularly the same assemblage of about 10 species that you find. 2 species. Sorry, I'm a little bit ill from the field mission. Still, it's cold here. We have 2 species that are living on sand, on the playa, on the beach, pure sand, white sand. Usually you go there and see nothing. If you are scuba diving there, you find little caves in the little holes in there, and in every one there's one of these fish. We have done research about the density and the population in one colony. They are appearing in colonies. You just take squares, count the fish, do an estimate about the size of this area by just scubaing around the edges and just pick up poles. And then you measure the size and you multiplicate, and you end up with a number of 1.5 million fish in one colony. 1.5 million. But then you have about 100 kilometers, not a single fish of that species. And we found 2 species living in rocky areas. Pure rock. No sand, no driftwood, no nothing, just rock. They're just hiding in the little scritches in between. That's their way to live. So you can say, basically, this group is specialized on the same habitat because 90% of them, roughly 90% of them are living in leaf litter. And we're just monitoring the leaf litter species. And they're independent from the type of water. The use of the habitat of a species is to know the use of a habitat of a species is very important if you have a background what happens when the climate changes. And we usually find monospecies situations. You have a river, a little quebrada, and you go there, collect fish, and you find just one species in there. You go for about 100 meters, you go for 10 kilometers, it's just one species there. Not a single specimen of another species. Then you have situations where you have 2 or 3, maybe 4 species, which is rare. This time we had one situation with 6 species, which is absolutely exceptional. And surprisingly of very different kind, that was something new for us. And then you may find situations where you have a single pair somewhere and no others around for hundreds of meters. And then come to the next spot, there's another pair. So pretty isolated. And what was unknown until 1991, when we observed it for the first time, Apistogramma, which are obviously fish that are ground orientated, are migrating. And they gather in bigger groups, a few thousand fish to a few 100,000 fish, and they are migrating, and for me it was surprising, during the times of falling water upstream. Usually you would think, well, water's lowering, let's get down. It took me about 20 years to get an idea why they do this. Swim against the current. Usually, the water is set free from little forest quebradas, gathering in the bigger quebradas and then in the big rivers. The areas that keep the water longest during the dry season are the little quebradas in the forest. They fall dry, but usually there is some pools that are filled with water during the whole dry season. The main rivers may dry out. I've seen that last year the Rio Nanai was completely dry. We walked through it, no water in it, in August, late August. So this is a very interesting aspect, and very rarely you find single specimens that are migrating. It's very difficult to determine if this specimen is just hunting or doing something, or if it is migrating. We have invented observation techniques to follow a single specimen for hundreds of meters, and if a single fish of this group is swimming a few hundred meters, we would say it's migrating, it's not just going for food. Usually the territory to pick up food is about 10 square meters, not more. So very interesting aspects that you have to follow. So the ecology is pretty clear. What is another very important aspect is the monitoring of populations and species. Well, we have here areas. Oh, there's something. Oh, okay, that was a wrong slide in there. The monitoring. Yes, I don't know here. The monitoring of species in some areas is very interesting. If we have in the Amazon, we have 130 species, roughly, more or less. In the ornamental system, pretty close to 30, and the Paraguay has the pity to have only 10 species at the time being. I guess that will not be more. It's roughly what is able in the Paraguay system. And we are monitoring an area here in the western Amazonian lowlands, and that is pretty interesting to see what is happening there. You have a group of fish that is pretty close related. This is what we call, in the widest sense, the Nicene complex. The setting up of the complexes is the result of morphological research run on a cluster analysis system. Therefore, we get these trees. There is a certain set of markers that we just take on every species on a certain amount of fish, usually 20 to 50 fish. We just check it, put it in a matrix, and run it through a CSS database system. And the result is just like this. Some of the species that are here called, like, for example, Apistogramma species pantalone, have scientific names meanwhile, but that's not important for this here. What is important here is that you obviously have fish that are looking alike in a certain way, and that they are all restricted to the area here between, let's say, Iquitos, roughly Iquitos, and Requena south. They even do not reach, well, the area of Tarapoto. Not even there. And very interesting that these fish have usually areas of distribution that are extremely small. About 50 kilometers in diameter, or only one little river. We now have one species that is living in only one pond. I wouldn't really call it a lake. It's about 1 hectare big. It's Apistogramma sinilabra. It's locked in one lake. We have collected the whole area around it very intensely. Every quebrada that we could get in, we collected, and we did find species, a lot of species, but not that one. So, what they do have in common, here they have certain blue markings on the body, the males, but if you look here, the females are all yellow, have black markings, but you can distinguish if you know about it. For example, this here, Apistogramma banshee, has black bars. That's easy to figure out here. But if you look on the species here on top, you have 3 species where the females have a black spot on the side, and here you have to look a bit more closer. And here there's another one like that. Here they get a broad black band on the side, Apistogramma nisoni, and here there's Apistogramma panduro. The band is much narrower and has another position on the body. These fish usually have a problem when they get mixed in the aquarium. You may interbreed it. You would think they don't. They're clearly distinguished. What is the most important thing is you have very small populations in some cases with very restricted areas. On the other hand, you have species that are widespread over the whole Amazon. There's Apistogramma agassizi. You can find it from Pucallpa to Belém and the mouth of the Amazon. One and the same species at the time. Maybe it's a supercontinent. When we look on the ecology of these fish, we have, well, again back here, I want to show you one species here. This was Apistogramma nisoni, the one here, down here. This was a species that drove around a lot of people for a couple of years because they took it to the aquarium, bred it, and they got only female offspring. Hundreds of fish, all female. When a new import came with new information about the ecology, the research groups and hobbyists started to breed this fish. It worked fine, super. With one problem, they got hundreds of offspring, but they were all male. As a biologist, if things happen like that, you would think there is something wrong. There is a problem. But what? What is the problem? You just keep this in mind. And I want to show you the importance of this problem a little bit later, just to keep you updated on that. Keep you thinking. What we also have done is some socio-biological biological research. We were interested in figuring out what is the life strategy of a single Apistogramma in the field. We did this in the Brazilian Amazon about 20 years ago now. I guess I get a little bit older now. And what do we have here? We just have a scheme of a river, of a river bank. You have here the altitude of the water is 0, and the river gets deeper here. Usually this shouldn't be blue, this should be brown, because this is soil. But to make it easier, we just left it like this. What's in the matrix? Usually, water has a certain structure of temperature. On top, you have the warm water, which here, in this case, was about 28 Celsius. Then you have an area that has moderate temperatures, about 25, 26, maybe. You have a real a jumper's ligament in there, so the temperature falls a little bit. And here you have temperatures in the deep water pretty close to 23 Celsius. If you walk into this river, you notice, wow, pretty warm here. And then you get down into the water, wow, the toes are not so warm, there's something going on, and you go really deep in, it's cold, you're freezing. You feel the cold water underneath. So, then we had a close look, we measured the temperatures, and then counted the fish. What do we see there? Well, we didn't only count them, we just trapped them. And extracted them and counted them. And you have a strip from the edge of the river that is very warm. You have usually small fish, maximum medium-sized fish, and you have a lot of them, indicated by the dots here. You have a lot of small fish. In the medium area, you have something very, usually not sexable. You can figure out if it's a male or female. You can't see. They're so small. Here in this area, where the temperatures are about 26 Celsius, you find some big males, adult males in the best condition, you know. So, the ones that the females would really like. And you find half-grown females that have started reproduction, and you find, very interestingly, you find a certain amount of males related to females. The relation usually is about 1.5 males, 1 male to 5 females, or 1 male to 10 females, about that. They are all reproducing. The females all are reproducing them. It is a very important part of the reproductive area of these Apistogrammas. And then you find, in the deep cold water, you find a completely different situation. You have a lot of big, very big males, about the size that they can ever grow up to. Maybe 8, 9 centimeters, really super-sized males, and very few, very big females. And we observed the behavior, and here in the deep water, the behavior is usually different. Here, males monopolize a territory and allow the females to get in there to make use of it. So they are the masters of the process. A female that they don't like, they push around and chase it out. A female that is not liked by the male has no chance to get into that territory. Well, I see some of you are smiling. Biology is always about sex. It's normal. So, in the deep water, the situation changes. Here, the males cooperate. They are holding a territory around the female's territory. A female here usually has about 20 fry, maybe. Here, up to 100, 120. These are the mega spawners, if you can talk about a mega spawner, if you talk about Apistogramma with 125. But this is mega spawners. They are very important for the population. But the risk of predation there is, of course, much higher. This is the area where the big cranicycla live, where the big catfish live. The risk to be eaten there is super high. So they have only a very short time to produce there, and of course transpose their genes into the next generation. Well, what we don't see here is fish. of medium or small size, and we don't see it here. But we find it here. So obviously all the fry is leaving this area and going into this area. Usually the fry, when Apistogramma guards the fry. The females watch the fry, they lay the eggs are covered. The larvae hatch about 1 or 2 days after spawning and are transferred by the females to different locations, depending on the pressure of predation. Then, after about another 6 to 8 days, they become free swimming. So it's about 10 days that they are not capable of doing what they want to do. They are depending from the female's behavior completely. After these 10 days, they could swim away. But the female and the males pick them up and put it back into the little group that they are guarding. The brood care usually takes about 20 to 30 days, which is a very interesting time frame. 20 to 30 days. Keep it in mind. The sex ratio that we observed in the field here was, in the deep water, 3 males per 1 female, maybe 5 males to 1 female, which is the extreme opposite of what we have here. And if we take this fish here from this area, from this shallow warm area, and we raise them in the aquarium, we get a sex ratio of 1:1, which is ideal in biology. Well, 1 female, 1 male, ideal. Well, one may think. Well, I know that there's other opinions about that. But here we come to the climate effects. If we look at this, what happens when climate changes? Well, before that, we have to know a little bit about the general climate there. Apistogramma are capable of surviving temperatures between 12 Celsius, which is very, very low. They would survive Lima today, and 35, which is Pebas last week. So, forest creeks are usually having temperatures between 20 and 28. The main rivers between 24 and 30. What was a little bit surprising, you usually think during the rainy season the water temperatures go low. No, they go up. In the Rio Negro, for example, the average temperature of the water in the rainy season is about 5 degrees higher than in the lower water season. Maybe, of course, the bigger water body is more capable for the sun. Lakes are different. They can have extreme temperatures. We have one measure that was about 35 Celsius. And backwaters, of course, well, they may be even higher than 40 Celsius, but then all the fish are there. Based on studies on reproductive biology, we have some very interesting aspects. Sexual dimorphism in Apistogramma is very, very interesting, but we have some reversed. And here we have, of course, this background. This dimorphism enables the fish to notice, very easy, who is who, and on the other side, what sex are you? And you put an Apistogramma female with a group of males in an aquarium with a bottom that is straight up, and you put a different temperature strata in there. We did it in a very big aquarium, about 3 meters long, about 1.50 meters high, and sealed it in, and put little caves there, just like on a ladder. The females just prefer that ones that produce the sex that is needed. If there is many males in the tank, the females go very low down in the aquarium to the low temperatures. And now we come to the very important point. Low temperatures, by experiment, produce a lot of females. Temperatures about 20 Celsius, 23 Celsius result in offspring that is usually about 90% female. If you are at the medium temperatures, about 26, you have the biased sex ratio, which is 50-50. If you go to 28, 29 or higher, it may result in unisex male offspring. This is done in research on at least 33 species of Apistogramma with about 6,500 experimental sets, which is a lot. In these experiments, only that sets were counted where the number of offspring that died between the egg laying and the adult size after a half year when you can sex them was less than 10%, just to make sure that it was not an effect of selective mortality on males or on females. If you have only 10% loss, this is excluded. It's just a mathematical problem. But a biological problem, of course, too. This is very important, of course. This explains what happens in the field, potentially. But we didn't find these results in the field. If you remember this matrix, the sex ratios were different. You have two different sex ratio systems. You have a genetically driven sex system, determination by temperature, and you have a sociological sex ratio, which is a complete different thing, which is driven by dominance and aggression and other things. And the diversity of Apistogramma is driven by something else. The sex ratio within a species is driven by temperature and socio system. And here we have another example of social system. For the speciation process, it is very important that you have an isolation between species. This is not necessarily a genetic separation. It may be a behavioral one, a pre-spawning barrier. In Apistogramma, the colors are playing exactly this role. How? That's a very important question. Well, it's pretty easy. If you're going to a discotheque on a Saturday evening, well, usually the girls dress up, the guys try to have a Porsche to drive there, so to impress the other one, which is easy. Normal behavior. Well, in this sense, these little fish are pretty human, because they do the same. They just dress up. If they are ready to spawn, the females put on the yellow color, and if you present, let's say, you know Lego, this, yeah? If you take a Lego, and a yellow Lego, and put black spots on it, and you present it to an Apistogramma male, it will react at once. Display, you know? It works. A little crazy German researcher, a colleague of mine, has produced a machine on an aquarium that is taking around this Lego in an aquarium and through an invisible to invisible part, and the males can follow it. And they are just swimming around like that, when then vanish, and then it appears here, there, the man chasing there, just to get to the female, so the potential female. For a male, only the color counts, the form is not important, yellow and black. Well, there may be some men that are blind like that too, I heard about that. So, well, but in Apistogramma, the males are the problem, of course, but the females making the choice, because they are much more picky than the males. We presented, well, how to figure out who's who is important. First, a female has to figure out which species it is to go to reproduction. Yeah, for the reproduction, they have to invest a lot. First, they lay the eggs, which is costly. It costs yolk, fat. The orange stuff in the eggs is just fat, and well, the water will rinse in anyway. They have to invest time, and so they have to guard the fry. They watch it. This is taking energy. They may have to take risks to defend this fry, and to be able to that, these females may use another strategy. They may just go ahead and take a male and involve it in the brood care, right? Just come in, join in. We may have more offspring. We have more success if we do it together. Well, and of course, there may be another strategy. We have now one species that is mouse brooding, which was a big surprise, but expected. And of course, it's the biggest species, because if you want to do mouse brooding, you have to be big. Well, this in mind, we get the temperature-dependent sex determination here. Of course, it's a little bit dependent from pH in some species, and it is always like this. And what is very important, here is just the result that I told you before. What is very important, you can reverse the sex. The females have chosen the situation. You expect a temperature of 23 Celsius, you expect males. No, females. But you take these eggs and put it to 29 after a certain time. Then you have another amount of females and males. So it's clear, it's not genetically fixed. It is just dependent from the temperature. And you can, dependent from the time, you can produce different results. So finally, the sum of temperature degrees that is collected during a time of 30 days is decisive if a fish is a male or female or not. We can discuss it later if you want. You do it the reverse way, the same result. We have one case now where the sex has been changed in an adult specimen, and we can prove that this specimen really has been a female before it worked as a male. Of course, we have a photographic report on that specimen continuously. We have the specimen preserved, and we have cut the gonads after fixation. And we find they have ovaries, ripe ovaries in there, and they have ripe sperm in there. It's completely crazy. So temperature effect is not restricted to fry, but may also be seen in adults. And why do they have this reaction to temperature? Well, if we are looking on it, they survive temperatures that are pretty low, 12 Celsius, and they are active at 18 Celsius without any problem. Then one comes, of course, to the idea that something with the last ice ages may have been influencing it. There must be something connected to it. And of course, you know Hafer's refuge theory, that during the ice age, some plants, some butterflies, some other animals survived in some forest refuges that were left over. Of course, the sea levels were very low during the last ice ages, I guess 300 meters lower than today, which is a lot, 300 meters. And so there were no flooded areas in the Amazon lowlands because the water just drove up in the Atlantic. And so only these dark green areas here, or the black ones were the central refuges, and the green ones the wider ones. And if we plot this on a map in Apistogramma species, we have a congruence there with species richness and types of fish. We have some pretty interesting plots there. Here is the number of species plotted. The biggest one here is the Rio Negro with 25 different species. Wow! Just a result of our research. And if you look on the map, most of the species-rich areas are connected to these refuges that Hafer has researched by drilling in pit ponds has shown that this is not completely right, because the forest areas may have been much bigger than postulated by Hafer, which would make this scenario fit much better to reality than the others. And what will happen when temperatures rise is pretty easy to see. We have had an effect on the sex ratio of Apistogramma. Sex determination is driven by temperature. But why? This is artificial data. It's a rough estimate of what may happen. If you look on the temperatures in fish, you have a dependency of external temperature in the biochemical reactions. There is a chemical law. If you higher the temperature about 10 degrees, you double to triple the speed of reaction. And if you have here the metabolism in the background of a fish, and you look at here, the daily energy expenditure just to survive is the blue line here, depending from temperature. A fish has a certain time during the day to take in food. He has some other things to do. He has to produce offspring. He has to defend the territory. We just exclude this from our idea. We just look on the point, what do I need to survive? The minimum, basic metabolism. And you have a straight line depending from the temperature. And then you just balance it to the amount of food that the fish can take in during that time. If the temperature is very low, it's very easy to take in enough food to supply yourself with the amount of energy that you need to survive. The higher the temperatures get, the more difficult it will be. And if you look here, based on the data that we took, we come to a point here about 32.8, I guess it was, Celsius, where you have an equal. So here, the fish can just take as many food as is necessary to survive. If it's getting warmer, the fish will starve. Even when it's feeding, it's starving. It will die. Sooner or later, it will die. It's just a question of time. But if you add up all the activities that a fish has to carry out during its life, reproduction, swimming around, picking up food, fighting with other females for the fry, interaction with the males, or the males with the females, whatever, the expenditure is much higher. This curve gets steeper, much steeper, but the amount of food that the fish can take in is even getting lower, because the time spent on other activities is limiting, of course, the time for food intake, so it's getting steeper. So they will die at maybe 30 or 29 degrees Celsius, which is the next problem. In a normal environment, in a normal forest quebrada, that's not a problem. The temperatures are about 23, 25 Celsius. That's normal for Apistogramma. We have some situations where we found females breeding on 30 Celsius, but that is only a short time during the day when you have a little river exposed to the sun. The temperature is falling very rapidly after dawn to temperatures about 25. Super rapidly. But 2 hours later, the water is cold. So everything is fine. If the average temperatures in the environment would go up, this changes, of course. Well, this is not the end of all, but if the temperatures fall during the night, it's fine. If the global warming would just set up the temperatures, let's say 3 degrees Celsius, just 3. The fish get into 2 different traps. First of all, they have energetic problems. They can't eat enough to run the normal process. For females, the problem during their ontogenesis is to get enough food to develop good gonads. If they can't make it, they just switch to another program. They just switch to the male program. So if there is problems, the malfunctional system will be put out and just switch to the poor male system, which doesn't need so much energy. The testes of fish are usually very small compared to the ovaries, super small. It's a factor about 100 in these fish. And if the temperature gets higher, they have this energy problem during development. And the next step, of course, if they really make it to a certain age, they have problems because they need more time for food and have not enough time for reproduction. The time is reduced, so the number of offspring may sink, may get lower, so that the dominancy of a species in the system may be taken off. It is very, very interesting. There has been a publication about 1.5 or 2 years ago, revising sex delimitation systems that have been published in literature. And they just came to the point that climate changes may not have any influence on fish populations. If I monitor the data that we have, I have serious doubts if this is really the case. We have these 2 problems, and there is another problem. The extremes in the water levels in Amazonia, as it is expected, are getting more intense. You get higher water levels, you get lower water levels. And this system is just wiping out maybe complete populations of fish that may produce genetical gaps between populations, and it might have a completely different aspect on speciation than we might expect. It may lead to higher fragmentation of fish populations and to more rapid speciation. That might be one effect of climate change. But on the other hand, we may lose the species. That may be the other problem. We don't know it at the time being. This is point of discussion. Yeah, and I guess this discussion needs more food from research in the field. And I can encourage you, just go into the field. Take a thermometer, take some net, catch some fish, measure, do all the research that is necessary. Don't stick to the laboratory, please. Yeah, because you see, if you're working on this, it's to the most interesting part of biology. As I mentioned in between, it's all about sex here. So you get to the interesting parts. So, thank you for your attention. I am here for any sort of questions, discussions, whatever you have. Thank you.
¿Hay alguna pregunta?
¿Puedo preguntar en español?
Sí, claro.
¿Ya? En el caso de estos peces, de los, ¿cómo dijiste que era?
Apistogramma.
¿Es posible encontrar algún tipo de que puedan producir huevos en estado de diapausa?
En Apistogramma es no. Son otras especies, por ejemplo, Rivulus, con huevos en diapausa. Sí.
Claro, lo que pasa es de que me llama mucho la atención con relación a que hayas encontrado áreas muy pequeñas con especies, o sea, me refiero a que esté muy aislado. ¿Y qué pasaría? O sea, ¿qué pasa si el área se extingue?
That is possible. We have this case. Yeah.
Yeah, because I noticed, for example, that in one you talk about maybe in, I don't remember this.
CD Lava.
Ah,
Yeah, in the little culture.
exactly.
Yeah,
What happened with that?
it will die out.
And so that means that maybe it's more species than exist in the past, for example.
It's difficult because it depends on the speed, how fast the genetical barriers are set. If we have severe climate effects, we will probably lose these isolated species that are isolated now, but on the other hand get some completely different other species. And what is also possible is that they may shift in their sex ratio by selection to higher temperatures. But there is a limit. There's a physiological limit in vertebrates in the water, usually about 35 Celsius in freshwater systems, that they can skip for a permanent time. So we have some cases in African lakes where the fish can stand temperatures about 55 Celsius, but only for a few minutes. And if we have these high levels, we may get problems with the sex ratios here. So that will trigger the breakdown of the populations, possibly. The number of species, all in all, may be stable, but we have a completely different system of species. It changes completely. That is a problem. And the species will be small. The fish will get smaller. In lower temperatures, you have bigger fish than in higher temperatures. So that is a problem in bigger fish species too. If you have bigger catfish, they have the same energy problems. And if we are fishing big catfish for consumption, and we can't get big ones anymore, that is a problem. Because the population in Amazonia will grow. If I monitor what happened, for example, in Iquitos, in 1970 something, I read in the paper, the population of Iquitos was about 45,000 people. Now it's pretty close to 750,000, which is pretty close to 20 times as much. Incredible. And it's still exploding. The population is still exploding. And this happens to other Amazonian cities too, of course. It's just an example, one out of hundreds. Yeah, that's it.